Okay, so in Blackboard, on our main page, when you scroll down, you'll see, and as you guys have already experienced looking at the stuff before coming to class, hopefully, dilution could be quite a monster, right? So I have a whole separate folder just for dilution, and I have a take-home quiz for you guys to do, as well as a whole packet that I already gave to you guys last week. So I'm going to go through in my lecture. It's just not opening. This is that handout you have already. The last page is for you to do as homework, and the answer key is right here. So I expect you to do it and then check your work. In addition, this is an old recording. I'm going to try and do a new recording explaining that homework to help you out with that. And then this is just another sample problem. Like if you have trouble with that first one, then do another one. Okay, so here is that one, and here is its answer key. And then this is the take-home quiz that I've already given you, right? And later there'll be a link for that. And then I have even more practice if you need it, as you can see, right? So practice, practice, practice. If you can't do all these practice problems, don't take the quiz. Come see me, right? And let's go through it, make sure you understand it before you do the quiz. If you feel good before next week, just make sure you enter. There'll be another link here I'll put up for you to enter the answers to that quiz, and it does count as a quiz. And then you'll still have a quiz on dilution next week in class, right? So we're going to do this a lot. And then it'll also be on the first part that, actually what we finish up today, which is the Kirby-Bauer sensitivity, which is what I'm going to lecture on first, really quick. So all that stuff in that folder is related to dilution, where today's lecture, which also includes antibiotic susceptibility, is going to be in the Lab 7 folder, as well as the recording that I'm doing right now, actually. So let me go back up to the beginning. Did you have a question? Ugh, it didn't go. I hate that. I didn't click on it. Did you have a question? No, I want you guys to do it online, because that way it's going to give you the answers, right? And it's going to be in that format that, you know, you can't cheat necessarily, right? You're not going to see the answers until after you put them in, where, you know, you could look at the answers ahead of time, right, when you're doing the practice problems, but you're only shooting yourself in the foot if you do that too. And especially if you don't understand how you're getting to those answers, right? So today we just did antimicrobial susceptibility test. Sometimes it's referred to as sensitivity. Those two terms are kind of used interchangeably. And we used a particular method. Just remember how when we did the staining, there were certain methods or different methods out there. So this is one particular method for antibiotic or antimicrobial susceptibility, and it's the Kirby-Bauer. And then we set up a standard plate count, which is also sometimes referred to as a viable count, because only the cells that were living are going to actually grow, and that we're going to actually be able to count the colonies that arose hopefully from one or a few cells. And we'll talk about that problem with that. So I'm going to go really fast through this PowerPoint and not go over most of the detail here because of a time issue. So what you see here is a plate, and you see bacteria growing here, and this is a fungus, a friend of ours that we know, penicillium. And so this one produces a chemical under certain conditions we know as penicillin. And it's producing it here, and it's diffusing into the agar that both of them are growing on. And because of that, this type of bacteria is inhibited by the chemical that's diffusing out into the media that you see here. We call that area a zone of inhibition, where we're stopping the growth of the bacteria due to the antibiotic. Well, instead of putting, you know, the organism onto our plates today, we actually put discs that have a set concentration and amount of a certain antibiotic. By the labeling you saw, different ones had P and S. Those were to denote the different antibiotics that we're using. It's a coded system. So two important terms. Antibiotics, right? This term is sometimes improperly used. The true definition of it is a natural antimicrobial agent produced by microorganisms. So this is chemicals that they've been producing, as you see here in that plate. This is a natural thing that happens. We've basically just stolen their technology, right? We've stolen their chemicals and used them externally. So microorganisms actually produce these to fight other organisms. And they're fighting over space and food availability. The same things people fight over, right? We're not all that different. Now, antimicrobials or antibiotics include antibiotics, right? They're a general term, right, that includes everything. And so these are all substances, both natural and synthetic. So some things are actually been made in the lab, right? Been synthesized from maybe other organisms like plants, right? And they kill or inhibit the growth of microorganisms or viruses, right? Because we have some antimicrobials now that work against viruses. Then we use the term inhibit or kill, right? Some of them can't actually kill the organisms. Some of them just inhibit their growth. This in our bodies gives time for our immune system to then kill the organisms, because these guys grow much faster than our own cells. So sometimes they can overwhelm our system before it's able to kill off the bacteria. And then sometimes, we're not able to do that at all. And people who have compromised immune systems, they're never, if you give them a bacterial static, which is an inhibitory drug, static, think about staying still, right? Then it's not going to be enough for them because their immune system can't do the second half of the job. Bactericidal, when you see it end in C-I-D-A-L, means to kill, right? So these are antibiotics that kill. And we've used an example of both of these today. So we've used chloramphenicol, which is bacterial static, and tetracycline, which fall under the inhibitory. And then we're using two in this group that we collectively refer to as beta-lactams, ampicillin and penicillin. Penicillin is very similar to the natural form. Ampicillin, anything you see that ends in cillin, is a chemically modified penicillin. So we would refer to this as being semi-synthetic, right, where we've actually modified the penicillin so that it works a little bit differently against different organisms. Aminoglycosides is another grouping of the ones that will actually kill. The one that we're using in that group is streptomycin, and ciprofloxacin is another one that would actually kill. You can see next to these cute little cartoons more categories and more types of antibiotics that can either kill or inhibit. So we have an example of each. So how do these things work? How do they inhibit? How do they kill? Right? It depends on the drug and it depends on the organism. Right? So in this table you'll notice that chloramphenicol is effective against the two organisms we're using today, which is E. coli and Staphylococcus epidermis. The reason why we chose those is because one's gram positive and one is gram negative, right? So we know structurally they're different, right? So gram negative have that outer membrane, right? And that's an extra layer of protection for them, right? It tends to make it really hard for antibiotics to get in. Whereas staph, they have just that peptidoglycan layer. They don't have an outer membrane because they're gram positive. Okay, so that distinctive difference between the different types. And then how the drugs work. So notice these are the main targets of how they work. So chloramphenicol actually interferes with peptide bond formation during translation. So that's going from RNA to proteins, right? So it's interfering with this step here. Others interfere with DNA replication, right? So interfering here. Or others are going to interfere with from DNA to RNA, right? And we talked about ones from RNA to proteins. And then even some proteins that are utilized for metabolism. The specific drugs interfere with those mechanisms. Or particular metabolic pathways, right? Where we go from one particular substance to another. So probably the easiest one, your sulfur drugs interfere with the folic acid pathway for making that vitamin in most bacteria, right? They're interfering with a crucial enzyme in that pathway, and it stops them from being able to make that nutrient. So that's what we mean by metabolism. And cell wall synthesis, so what specific molecule are we interfering with if we're interfering with cell wall synthesis? Them making this peptidoglycan, right? So that's the target there. And then we can even affect the plasma membrane, okay? So notice penicillin, it affects the peptidoglycan, and because of that, it's more effective against staph than E. coli, because remember, staph is gram-positive, so it's got that really thick, and it's really dependent on that really thick layer of peptidoglycan to maintain its cell integrity. So then over here, notice resistance. So lots of different ways. Poor uptake of the drug. Like I said, it's really hard for penicillin to even get into the bacteria to interfere with these pathways, right? Most of the time, all these drugs have got to go inside, right? Because all these things are happening inside the cell, right? So if the drug doesn't go inside, then it can't affect the target. If the target that it usually binds to has been changed, it's been altered, it can't bind to it, right? So there are lots of different ways. Some of them produce enzymes that actually destroy this antibiotic. They're called beta-lactamases because they destroy that structural component that we'll look at in a moment. So gram-positive versus gram-negative. So as we said, right, gram-positive, this would be our staph today. Very thick peptidoglycan layer. Gram-negative, that outer membrane really helps protect sometimes the antibiotics even getting in. And that's true for penicillin. Penicillin does not work against gram-negative. Straight up plain old penicillin, right? It can't get in. Poor uptake. And that's a natural born resistance for gram-negatives. So some other ones interfere in different ways. And notice we have here modified penicillin. That's what ampicillin is. So notice we can use it against E. coli, right? The chemical modification that's happened, it can get in and it can interfere with the peptidoglycan synthesis for those bacteria. And we'll see that. And we're going to use this drug later on in the semester. These two work similarly and that they're interfering with the ribosome, so we're talking about protein synthesis, right? So resistance, as I said, the drug itself, when it comes in, it's usually going to bind to some type of target molecule and it's going to interfere with that process, whether it be DNA replication, RNA, protein synthesis, some metabolic pathway, right? There's some interactions happening between the chemical and its target. So one way is that if the target changes, so notice that this is the target but its shape has changed, right? This can no longer bind to it, so therefore it's not effective anymore. So then that bacteria would be resistant. Okay. And the reason, right down to the molecular level, would be because it can no longer bind to the target. So for this one, sometimes the antibiotic will come in and then there's an enzyme inside the bacteria that will destroy it. And this is very common for penicillin. Notice penicillin's got this ring structure. It's known as the beta-lactam ring. There's enzymes out there known as beta-galactanases. Guess what they do? They snip this ring. And when they do that, it stops the antibiotic from being able to bind to the target. Now, when amino acids bind together, right, that's a reaction called as a peptide bond, right? And so that's why, again, remember there are amino acids in peptidoglycan. That's why it has that in the name. Peptido referring to the amino acid component, glycan to the sugars. Was there another reason why I wanted to mention this, Peter? That bond? That's what forms here.
The beta-lactam is
a peptide bond.
The ring, and it sort of mimics an amino acid in that peptidoglycan. So.
Right. Yeah. So it's very similar that you have. This is very similar to amino acids in the structure. So the other thing is that some of them decrease the uptake, right? So it just doesn't get in, right? And if it's not going to get in, it's not going to be effective. Other ways is that they literally, it comes in, but then they pump it right back out. They're like, oh no, that's bad news, right? And they like throw it right back out the cell. How they do that, I don't know, but we do know some of them do that, right? They can throw it right back out the cell. So if you have a population of bacteria, right, there could be different ones within that group. Genetically, they're different. Their target could have been altered. Maybe they figured out how to bail their boat, right, how to throw stuff back out. So when we apply the pressure of an antibiotic, the ones that are sensitive, who have not changed in any way, and that the antibiotic actually destroys them or inhibits them, they're going to be eliminated from the population. And then you're left with the resistant ones, right? And this selective pressure we commonly refer to as natural selection, right? Survival of the fittest. So then all you're going to left with is the resistant organisms. And this is what's unfortunately happening over time, right? As people misuse antibiotics, don't tape them properly, take them when they don't need to be taking them, the overuse of them in the environment, and the use in animal production. We're building and we're exposing so much bacteria to so many more. The selective pressure is there that we have a lot of resistant organisms. So one example that you'll probably come into contact with in hospital type settings is said MRSA, but it actually is MRSA, which stands for Myctogen-resistant Staphylococcus aureus. That was a very common penicillin-like drug that was utilized against staph infections. And because of that, we've got a lot of strains out there that are now resistant to those antibiotics. So now we switch to using vancomycin. So guess what we have now? Vancomycin-resistant Staphylococcus aureus, right? It's just a matter of time because they're constantly changing, right? So, and this is what drives the evolution. And the evolution, because of the use of antibiotics, is towards resistance, which is really kind of frightening. So, most of you guys probably have this at home, right? Triple antibiotic ointment. So why is it called triple, or Neosporin if you're not cheap like me and you actually buy the name brand stuff? What does it have in it? 3 antibiotics, right? That target 3 different types of interfering with cell maintaining its stability. So polymyxin B interferes with the plasma membrane. And because we have plasma membranes too, it can be toxic to our own cells, which is why you use it topically. You do not ingest this. The other is bacitracin, and this one can be toxic if taken internally as well. It targets cell wall synthesis. And then the third antibiotic, you can see where neomycin gets its name, right? Or neosporin, right? Is from the antibiotic neomycin, which interferes with protein synthesis. And if you actually read the label, right, for external use only, if swallowed, call PERS and control immediately. Because as I said, most of them can be toxic to our cells. Not supposed to take it internally. Just for minor wounds, right? Notice that not to use on certain types of puncture wounds, right? Actually read labeling, right? And follow the instructions. You don't want to put it on large surface area because then you would absorb a lot and it could damage your own cells. So when we're testing, right, especially in this world of resistance, sometimes you'll give antibiotics to a patient and they still have the infection. So they might do some antibiotic sensitivity testing next to determine which antibiotic will actually target the actual bacteria causing the infection. So one of the things they could do is, like we did today, create a lawn of growth, right? So they swab the whole entire plate and then they put antibiotic discs on that have a set concentration, and that's going to diffuse out and hopefully create these zones of inhibition. And what you're going to do next week is you're going to actually measure those using a centimeter ruler in millimeters. So each one of those little lines is a millimeter. And in this method, there's a table that comes along with it, right? You probably saw in your homework. And you're going to compare that to determine if it is truly sensitive or resistant. It has to be a certain diameter. Now here, notice it did not, and actually here, no inhibition of growth at all, right? It's clearly resistant to that antibiotic, right? You definitely would not give that one to your patient. The others you'd have to measure and compare to the table, right? So these clearing zones are zones of inhibition. And I am going to skip through these other ones because of time. Maybe I'll do another recording later. So how does the antibiotic get from the disc to the agar? It just simply diffuses, right? But it's going to diffuse out and down into the agar, right? So that's why the depth of our agar is important, and that's why there was a set amount of agar in your tubes, right? Because if we were to pour it too thin, then it would affect our diffusion rate because it wouldn't be able to go as far down. Does that make sense to you guys? Right? So we're expanding down as well as out when they diffuse. Right? So it's going to be radially as it goes in a circular fashion, outward but also down. So diffusion happens. To ensure the reliability, as I said, we made sure that they were a specific depth for us. And so how would this affect it though if they were too thin? So think about it. They can't go as far down, so they're going to go further out. And we're measuring that distance out, aren't we? So is that going to affect our results? Absolutely, right? It would really throw off our results and make them appear larger than they should be. And then, so Peter actually calculated it out. The pores. He loves the math. So
Mine is 25 mils.
25 mils were in that to create, based on the dimensions of the discs, the Petri dishes we use, that should fill it up to 4 millimeters deep. So the other thing is that we prepared those fresh today, right, in lab today, and there's a reason for that. As you sit them in the refrigerator and stuff, depending on how you keep them, it'll affect the moisture content. And does moisture affect diffusion? Yeah, so if they're too dry, it's not going to diffuse as far, right? And if they're too moist, then it's going to diffuse further, right? And you can very simply demonstrate this if you just take a wet paper towel and a dry paper towel and you put 1 drop of dye, you'll see that the dye will spread out further on that moist paper towel as opposed to the dry one, right? Because that moisture is going to help spread it. So here the example they're saying that, so they don't give us a specific one. Suppose you specific time frame, free of visible moisture, right? So if it's too moist, it's going to be a larger zone, right? So, and then the McFarland standard, right? Another reason why I prepped it right in lab today. It must be used within 30 minutes if we're truly following the protocol. And the reason for that, for an organism especially like E. coli, in a matter of 20 minutes under optimum conditions, it would double, right? So you'd have to double the amount of bacteria. And so we want a pretty standard, but notice we didn't use the spectrophotometer. We kind of eyeballed it to that pre-made standard. That pre-made standard is supposed to be around 1.5 times 10 to the 8th, which set out would be 150 million cells per milliliter, right? And as Peter said, that's more cells than the population in China, you know. So, I mean, we're talking about a lot of cells, right? And it didn't even look that cloudy, right? It barely looked cloudy at all.
For a 10 mil culture, 1.5 billion cells in a 10 mil culture. That's what.
Oh, yeah, if you had a 10 mil culture. So as I said, we're using 2 representative organisms, mostly 2, so you can compare this difference between gram-negative and gram-positive. So remember, what's the difference here? Right? And it's the same difference that why they stain differently. It's the difference in the cell wall structure, right? That peptidoglycan. Gram-positives, the staph has a really thick layer. Gram-negatives have that thin layer, and they have that outer membrane. And that really plays a key role when we talk about antibiotic susceptibility here. Or sensitivity. I stumble on susceptibility. I can't say that word. Right? So there's our differences, right? And, right, so like straight up penicillin. So then next week you're going to interpret your results, right? So we're going to actually measure. So here's an example of that. If we had chloramphenicol and it measures 25 millimeters and penicillin measures 25 millimeters, notice these are both the same, right? But these antibiotics are not the same. They don't respond to cells the same. You also have to know what bacteria potentially you're maybe working with. Because penicillin's really only going to work on staphylococci. And what you're really concentrating on is it sensitive or susceptible? And so for here, it has to be equal to or greater than 29 millimeters. Is it? No, it's less than that. So it's this particular organism, maybe it's not even a staph, but if it is a staph, it's resistant, right? Because it's less than that 29. Now, for chloramphenicol, it only has to be 18 or greater. So in this case, is it sensitive or susceptible to this antibiotic? It is. So this is the one you would prescribe to your patient. So again, it doesn't matter, you don't compare antibiotics, measured sizes, you have to compare it to the table of known data, right? That's the key take home. And of course, I'm not going to expect you to memorize the data chart, right? I'm going to give you guys a copy of it on your quiz, on your test, right? So you just have to interpret as we're doing right here, okay? So, right, so chloramphenicol would be our choice. And then how does the antibiotic diffuse? Remember, it diffuses out and down, right? So all those factors come into play. After incubation, does the antibiotic extend to the agar past the zone of inhibition? Does it go past where we're seeing? Yeah, it does. It keeps diffusing out, but that zone of inhibition is at the concentration in which it's still inhibiting. Because think about this. As it goes out, it diffuses out, it's going to get less and less concentrated, right? So that area where you see the stoppage of growth and then the growth starts, that zone right there is the minimum inhibitory concentration. So it's diffused out, right, to such a low amount. That's the point at which it's at its lowest concentration and still inhibits growth, right? So what we call the MIC, minimum inhibitory concentration. So that's the zone, that's the area. It's still diffusing past here, but it's not stopping growth. It's too dilute, right? It's not concentrated enough. And so there are different ways you could do a serial dilution for this, right? Where you have a set number of bacteria and you dilute out the concentration of the antibiotic. And you'll see that there's still turbidity in these tubes, right? So they're still growing. And tube 2, that's the lowest concentration. It was 2 microliters per milliliter that actually stopped the growth of the bacteria, right? We don't know the concentration in the discs test because we don't. The test isn't designed that way, but there are other tests that are designed that way. For this particular test, we don't need to know minimum inhibitory concentration. We need to know if it meets that for that particular antibiotic, and we have that information in that table, right? This is a standardized test. You're not trying to figure that out, right? So we always reference our table. So next time, you know, we'll definitely look at, right, the antibiotic, and we'll measure our zones, and we'll determine if it's sensitive or resistant by looking at our table, right? So just as a quick example for streptomycin, when you look for streptomycin, it's got to be 15 or greater, right? For this E. coli sample, we measured 20, so therefore it's sensitive, right? Because it's 15 or greater.
Click at one form.
Oh.
Sensitive.
Yeah, see.
Out the chart one at a time.
Yeah. I know, it came up.
Okay.
It came up, right. So, and this one actually fell into intermediate mode. I'm going to prep this other gram-negative for us, too, to be able to look at next week. And you'll see that it tends to be resistant to a lot of antibiotics. And we'll talk about that when we look at those plates. So we could think through, right? This is a scientific test we're doing, so we can form a hypothesis, right? We run the test. We're going to get results, and we're going to talk about what those results mean, right? So we're going to be able to, as we saw, you know, is it effective? Is it not effective? Did we follow the steps properly? What questions are we asking? How do we measure our results, right? And then what does it tell us? So enumeration, this is the tough stuff, and I have like 10 minutes. Okay, so there's different ways of counting bacteria. We could put them in a microscope slide, a special slide where it has lines on it, and we could count individual cells, right? That's called a hemocytometer. We don't have a picture of it though, huh? I need to get a picture of that. Yeah, I've done this before. We could do indirect, where we do biomass, where we could use a spectrophotometer. We could measure particular products. There are some machines that can measure the level of ATP and therefore calculate about how many cells are in it. I've used methods like that in research. We're going to do, and we did today, the serial plate count. The drawback with this is that we're talking about growth, right? And so we got to wait time, right? So we got to wait 24 hours, right? We got to come back. We don't know today, right, what the number is. Where these other methods, right, we would be able to get immediate results, okay? The difference here, though, is that these techniques, unless you're using special dyes here or special products here, you not necessarily can tell between living and dead cells, and you might need to know that. Where for our test, was another reason why it's called a viable cell count, is that only the cells that were alive when we plated them are going to actually grow. But there's other factors that come into play. Did you guys notice that our plates were different colored today, right? It was 2 different types of media we're using. Are they going to respond differently to the, yeah, right? Because it's different types of food, it's a different type of environment. And there's a reason why we use those different kinds of plates. So here's turbidity, right? The light shines right through, gives us a really high, none of the light is absorbed by it. We get a pretty high reading on the spectrophotometer, where if you have a lot of organisms, they're going to refract or absorb the light, and less light is going to get through to the meter. We get a much lower reading. And so I had wanted to, if I had time today, is actually take our samples and get us a reading on the spectrophotometer back there. I'm going to have to set it up and do it another time. But as I said, we just eyeballed it today, and then we diluted. What? Okay. Okay, so I'm going to also skip over Peter's explanation of counting and like how counting came to play. So you guys can read through this on your own if you're interested in the history behind how we count. I'm going to get to the actual dilution rules at some point. Lots of examples.
I will just say that the English-speaking Europeans weren't counting really until about 500 years ago. So it took mankind a long time to figure out how to count. It's not an easy thing.
Yeah, definitely not an easy thing to count. So we'll be using scientific notation, so you'll notice I have an example of that in one of the packets that I gave you. And so basically you always want one whole number and then a decimal point. And then the rest of the zeros past the decimal point is represented by your exponent, right? And typically we only go 2 numbers past the decimal point. That's the word, decimal point. Okay, so that's how you would set up each one of these numbers for scientific notation. And we'll practice that. So you'll notice that the standard it stands for standard, or System of International Units, is actually a base 10 system, right? It's based on groups of 10, numbers in the 10 grouping, and so that's the same as exponents. So remember when we talk about fractions, because we're going to use fractions, that the top is referred to as the numerator, the bottom is referred to as the denominator, right? So A is the numerator, B is the denominator. If you do anything to the numerator like multiply, you have to do the same to the denominator. I always think of fractions as evil Siamese twins. They're stuck together, right? And whatever you do to one side, you must do to the other, otherwise they get really mad, right? That's the easiest way to remember how to handle fractions. Evil Siamese twins, okay? And you can write any number as a fraction, or the inverse of that fraction, which we refer to as the reciprocal. So multiplying by 1 doesn't change, right, anything. So those are some important mathematical terms. So here, right, when we're multiplying by 5, you just imaginarily put a 1 underneath this, right? And so 1 times 5 times by 10, right? And I don't understand the way you have this written. Oh, you just did the reciprocal. Yeah. Oh, divide. I didn't even see the divide. Right, you have to flip it over when you divide. Which we're not going to have to do for this. Oh, we do have to create a reciprocal. You're right. Okay. Yeah. We multiply. The reciprocal is just a fraction flipped, right? So remember when you used to divide fractions, you'd have to flip the second one. So 5 times and you flip the other one to be able to do it. And the same thing, you'd have a 1 underneath this 5. So it's 5 times 10, 1 times 1, so it's just 50. And then several examples of that. So why do we make serial dilutions though? Because believe it or not, there are too many bacteria in that original sample, as you guys will see even in our first dilution, that they're not going to be spread out enough when you spread them, that we're going to get individual cells by themselves to grow up into little individual colonies, right? Isolated colonies like we did with our streak plates. There's going to be too much bacteria, right? They're going to all kind of run together in what we call a lawn of growth, okay? It's not until we get further down in the dilutions that we're going to have few enough bacteria that were put on that plate that you're going to have individual colonies that you'll be able to count. So let's look at dilutions. So the first rule for dilutions is we have to know what the individual tubes are, right? And today we started out with 9 mils and we added 1 mil. So our total volume was 10, right? Let me get that up there. So 1 mil into 9 mils gives us a total of 10. So another way to think of this is part over whole amount, right? What's the part I moved? 1 mil. What's the whole amount I ended up with? That's 10, right? Your whole amount always goes at the bottom, just like when we're talking about fractions in general, right? Whenever you divide up a pie, right, into 8 pieces, all 8 would be your denominator. Your numerator would be how many pieces of that pie did you take, right? Did you get rid of my picture? No, that's for something else. We did that. We did that before. So remember, we're adding here to get that bottom. You've got to remember you have to have your dilutant plus what you added to it. What's that whole volume you end up with? Still with me? That's the very first rule for every single tube. And so for all our tubes, if you guys look at your sheet, right, it's all 1 to 10. And so is the example that I have here in that first sheet that I have for you guys. So let's do something different. And I think I have the one in here that we did to start out earlier. So let's see if you guys can follow the rule, right? So 2 over 18. Is that right? No, the rule is part over whole. So it's 2 over what? 2 plus 18, which is 20, right? Part over whole, right? 2 milliliters plus 2 plus 18, which gives us 2 over 20. Now the only problem with this is not in lowest terms, right? So then we would have to put it into lowest terms. I promise you guys we won't do that on any of our quizzes. I don't think. Yep, or tests. So if you don't remember how to do that, remember evil Siamese twins. We want to make the top 1, right? So you could divide both the top and the bottom by 2, and you would get 1 over 10. Okay, that's probably the easiest way, is divide by the number that's at the top, both the top and the bottom. Of course, math is great because you can do things different ways and get the same answer. So if you were taught a different way, as long as it works, that's fine. So this is similar to what we did before. Notice that we have a 0.5 into 9.5, right? So again, remember when you're adding up your decimals, make sure you line them up, right? So 0.5 and 0.5 plus 9.5 gives us 10. But again, we don't have a whole number here. And so in this case, I multiplied by 2, which would give me 1. And that gave me 20. When you're dealing with decimals too, sometimes it's easier to think of it as money, right? So this would be 50 cents, right? Which is half of $1. So you know if you multiply it by 2, you'd get a whole dollar. Some tricks of the math world. So the next one is, we didn't just dilute individually 5 tubes 1 to 10, did we? No, we diluted the first one 1 to 10, and then we took out of that. So that sample we took was 1 to 10 diluted, and then we put it into 9 more mils, so we diluted another 10 times. So as we go up, what do you guys think we need to do to those values? Do we add them, multiply, or divide them? We multiply them, right? We use the multiplication rule, right? So we're going to multiply as we go. So everything's going to be in lowest terms, right? And you just keep multiplying as you go. So tubes A, B, and C, right? If we were to calculate C, it's all a combination of what A was, what B was, and what C was, right? Whatever dilution factor each one of those are. You just keep multiplying as you go, right? So it'd be, in our example, 1/10, 1/10, 1/10. And then you can just add up your zeros, right? We've got 3 zeros, so it's to the power of 3. Instead of writing as a fraction, if you put a minus sign, that denotes that it's the bottom of the fraction. So that's why we wrote minus on the board, right? So notice the 3 different ways of writing it. I like to stick to writing the fraction so I remember I'm doing a dilution factor, and I'm less likely to lose that little negative sign. But you can do it either way that you feel comfortable doing. And after I have 3 zeros, I usually always write an exponent form, because that too, you're going to eventually lose track of those zeros, right? And it's a pain in the butt to keep counting those zeros. It's easier just to keep increasing your exponent. Make sense? Okay. So for this one, right, tube A is 1/10, but look at tube B is 1/5, and then when they did only a half a dilution, 1/2 for C. What is the total tube dilution for B and C? So which one it's easier to do B first, right? So what do we have to multiply to get B? A times B, so 1/10 times 1/5, right? Which gives us 1/50. Still with me? Okay. Now if we want to find out C, what do we have to multiply to get C? You got to multiply A, B, and C. Well, we already did A and B, did we not? It's 1/50. Why do it over again? We already know what that is. Let's just multiply by C now, right? So 1/50 times by C, which is 1/2, will give us 1 over 100, right? So yeah, you could, do it all out again, but you've already done to this step, right? You just got to multiply in the new one, right? So as you guys are doing these, right, just keep going. Oh, okay. Yes. So you can do it shorter. So the next one, so we've diluted out all our tubes. We've done the serial dilution of all the tubes, right? And figured out what that dilution is. Then the next step was we took from the plate, from the tube, and put it onto the plate, did we not? Okay. Did we take an entire mil out, an entire unit that we're working within? We're working with milliliters, right? Did we? No, right? We took 1/10 of a mil, right? So how much bacteria would you have if you took a whole mil out as opposed to only 1/10 of a mil? Is it going to have the same concentration? No, you're going to have more bacteria, 10-fold more bacteria probably in that entire mil that you took, right? Instead of that 1/10 of a mil. You notice how I'm saying it, 1/10, right? So it's 1 over 10. So essentially when we took our sample, it was almost like we did another dilution, right? Because we took a small amount out. Everyone still with me? So mathematically, how do we calculate that? So you need to determine that we need to know what that final dilution was on those plates in order for us to calculate are the number of cells, or CFUs, colony forming units, right? Because we're counting colonies, we're not counting cells. We don't know for a fact that each one of those colonies was from a single cell. We're assuming it might be, right? But it may not be. So what do they refer to it in our lab manual as? Sometimes it's also referred to
refer to it
I'm
as
very
total plate dilution.
total
But
plate.
The quote refers to it as
plated
the volume
dilution.
of the original sample.
Oh, volume of the original sample? Lovely.
Basically, my total plate.
Oh yeah, of your very original sample. Right. But I'm very specific as to what we're talking about, right? So this is the total dilution for that plate. What is that dilution factor? So to figure that out, right, your plated dilution is the dilution of the tube that you took it from, right, times the fraction of the 1 milliliter plated, right? So what is that, right? And it's 1/10, right? It's 1/10 of a milliliter that we did. Because our dilution is in fractions, what we're multiplying by has to also be in a fraction, right? Otherwise it's not going to come out correct. So a tube with a total dilution of 1/10 and you transferred 1 mil, in that case, how do you write 1 as a fraction? It's just 1/1. So it would be the same, right? Because you took an entire unit out. But that's not what we did, right? So 1/10 times 1/1 would just be 1/10. It would be the same as the tube. But what we did was 1/10. So remember the rule. The total tube dilution, which is 1/10, times the fraction of the 1 milliliter plated, right? So what fraction of 1 milliliter? 0.1, which is 1/10 of a mil, right? 1/10. So 0.1/1 times by 10 will actually give you the same thing?
Well, I just
1/10.
multiplied by 10
Yeah.
divided by 10.
10,
Just moving
okay.
the decimal place over, basically.
Okay. Yeah. But it's 1/10 times 1/10 like you see here. That one's confusing me.
I would like to point out that in practice, you don't really want to plate 1 mil of a dilution because it would take too long to soak into the plate.
Mm-hmm.
Practically speaking, you have to use smaller volumes, so that you're talking about a fraction of it.
Yep. So same thing here. If you had a dilution of 150 and you have 0.5, how would you say 0.5? That's how much of a mil? Half. So you could write that as literally 1/2, right, in lowest terms. So 1/50th by 1/2 gives you 1/100. Is that total plated dilution? Last one, you guys. Last rule. When we look at our plates, we're only going to count plates that are between 30 and 300 colonies in order to be statistically accurate. Once we count them, then we multiply by that number to get, oh, you go into a whole bunch of stuff here.
Yeah, I have 2 slides in the
Okay.
way, so you want to keep going?
So I'm going to go to an actual picture. Read over this stuff because I will talk about it next time. So notice here, right, the number of colonies are written on the plates. Which one is between 30 and 300? H, because it's 130. You take that number, right, to get your titer, your concentration CFUs per milliliter, you take your number of colonies by your total plated volume. So here it's saying for H is 10 to the 5th. You just multiply, put in scientific notation, and this tells you about how many bacteria, right, would be in a milliliter of our very original sample we started with. Okay, so as I said, I'm going to try and do some more recordings to go step by step through the math, especially for the homework page and the example page. I'm going to post some more recordings for you guys. Probably work on them tomorrow, and this lecture as well. Go through the examples, right? See if you can follow the rules, right? Do as many as you need to till you feel comfortable. And definitely come see me. I learn best from working with you guys one-on-one. Don't be afraid to come see me, okay? Even if you can't make my office hours, give me a call, send me a text, send me an email, set up some other time before lab next week, okay? I will be here. I am better.